I'm Rick Lifton, the president of Rockefeller University, and I'm delighted to welcome all of you to one of my favorite events of the academic year, the presentation of the Meister Greengard Prize, an international award that honors outstanding women in biomedical research. This is the 16th presentation of this award. Since 2004, we have celebrated extraordinary women who have made major contributions to biomedical research in diverse fields, including molecular biology, cancer research, immunology, biochemistry, genetics, biophysics, and neuroscience. Discoveries made by our honorees have been among the most remarkable of the past half-century made by men or women. And tonight, we're here to honor another extraordinary scientist, Dr. Xiaowei Zhuang, for her remarkable innovations that are illuminating fundamental processes in biology. Xiaowei was chosen by members of the prize selection committee, a group of esteemed scientists that includes 4 Nobel laureates. Tonight, I have to comment, is bittersweet as we remember the prize's co-founder, our incomparable friend and colleague, Dr. Paul Greengard, who died last April of this year at the age of 93. His death was a great loss for the scientific community and for Rockefeller University and our family. He was a force of nature, running an exceptionally productive laboratory until the time of his death. But Paul was also a warm friend with a sharp wit and sly humor, who was a source of inspiration to all of us. Paul Greengard was a giant in the field of neuroscience. His research shed light on the fundamental causes of neurologic and psychiatric illnesses such as Parkinson's disease, depression, schizophrenia, and Alzheimer's disease. And his scientific legacy is further notable for his more than 250 trainees who now populate the world's leading departments of neuroscience. When Paul received the Nobel Prize in Medicine in the year 2000 for his crucial discoveries about the mechanisms of signal transduction in the brain, he and his wife, Ursula von Riedingsvard, who's here with us this evening, donated his monetary share of the Nobel Award to establish tonight's prize to promote the recognition of great science done by women, which Paul and Ursula believed was not being appropriately celebrated otherwise. The prize was named in memory of Paul's mother, Meister Greengard, who died giving birth to him. Paul and Ursula's vision and generosity inspired Friends of the University to provide additional funding. And in addition to the prestige of this award, the prize includes a $100,000 honorarium. Now, before I introduce our guest presenter this evening, I'd like to share with you a short video about the prize that was produced by Paul and Ursula's daughter, Ursi von Riedingsvard-Grieve. Please enjoy the film. The Meister Greengard Prize is a real gem here at Rockefeller, and I think it's a consequence of a visionary idea and the very high standards with which the recipients have been selected.
And now Professor Paul Greengard.
The genesis of this prize goes back to the year 2000 when I won the Nobel Prize in Physiology of Medicine. The prize is called the Meister Greengard Prize because it was named in honor of my mother, who died giving birth to me, and it seemed that it would be a nice thing, since I wanted to do something about discrimination against women, to name it in her memory.
One hope from this prize is that women who are fascinated by science, who are thinking about careers in science, will see these incredible women who have been leaders in science, have been successful in science, and are recognized in science, and see these women and say, I want to be like her, and I bet I can.
Here in my lab, we invent advanced imaging methods with molecular scale resolution and genomic scale throughput to study biological systems. We're made of cells, and then cells are made of molecules. So we want to understand, in general, these molecular interactions. How do they give rise to some of the higher order behaviors of the cell? These imaging methods need to have very strong, what we call resolving power, because the molecules are so tiny, they're nanometer scale. So one of the things that we did is an invention of a microscopy method called STORM, which overcome the diffraction limits so that we could get nanometer scale or molecular scale resolution to really have a very crisp picture of what these molecular players are doing in our body. One thing that I would dream about is, I really love this direct visualization approach. Really seeing is believing. And, this thing that you, Guibera, like to say, one can see a lot by watching. It's this kind of obvious truth, but it tells you that what you learn by just directly seeing things is an amazing amount of insights that you can get. Now with that, I can get a molecular basis of life, and I also can see this unfortunate patient who has this disease is because these molecules went wrong this way. I directly visualize that. So if I could have that kind of dream scenario where every single molecule in our body, what do they look like, where they are, how do they interact with each other, and how do they together give us life, and what went wrong in diseases. They're all directly visualizable. That I would love. And I would just keep working towards that direction. And how long that would take, hard to say. When I got that email about this sad news, that Paul passed away, I thought, oh, that's so sad. I would have so loved to actually meet him and see him, not having a chance to communicate exciting scientific stories with him and to hear his sort of wisdom and feedback. I felt like what I want to do the most is to keep doing great science so that I won't let him down, right? That kind of thing. And also promote women in science, especially encouraging young scientists, young female scientists, to tell them that, hey, we can do whatever we want if we put our hearts and minds to. There's no reason to believe that if we are interested in certain things, we cannot do it. And then encourage those girls to get into science too.
I would like to introduce Billie Tsien, our presenter this evening. Billie is one half of the dynamic architecture team, Billie Tsien and Todd Williams. The couple's creative partnership has spanned more than 4 decades and has yielded a portfolio of public art projects that can be found throughout the world. Billie was born in Ithaca, New York. She received a degree in fine arts from Yale and a master's of architecture from UCLA. In 1977, she began working with Todd Williams, and the couple married in 1983. They founded their architectural firm in 1986 here in New York City. Since that time, Billie and Todd have committed their careers to designing centers for arts, culture, and academia that can be shared with the public. Their aesthetic is one of elegance and modernity, and much like our recipient today, they have a deep appreciation for the small details that inspire beauty in the eye of the beholder. Billie and Todd's buildings have become celebrated homes for art collections, vibrant spaces for civic engagement, and rooms for quiet contemplation. Some of these projects include the Asia Society in Hong Kong, the C.V. Starr East Asian Library at the University of California, Berkeley, and the Barnes Foundation in Philadelphia. Closer to home, they also designed the LeFrak Center at Lakeside in Prospect Park, Brooklyn. Billie serves as president of the American Academy of Arts and Letters and is a member of the American Academy in Rome, the Architectural League of New York, and the American Academy of Arts and Sciences. In 2013, Billie and Todd received the National Medal of Arts from Barack Obama, and just this year they were awarded the prestigious Premium Imperial by the Japan Art Association. They are currently designing the Obama Presidential Center in Chicago, which is set to open in 2021. It's our great pleasure to have Billie Tsien here with us this evening. Please join me in welcoming her to the stage. Thank you so much.
Tonight is a very tender night, because in the act of celebrating, we are also remembering. We celebrate Xiaowei Zhuang, and we remember Paul Greengard. Indeed, it is the act of remembering which brought this prize into being. Paul named this prize in memory of his mother, Pearl Meister, and thus we simply complete the thread when we also remember him. His achievements and generosity in establishing the prize with his wife Ursula are known. But what I did not know until his memorial was the intimate and quiet generosity that expressed itself in the nurturing and mentorship of all of his postdocs. Person after person stood up and talked about how much he meant to them. And of course, in many ways, the strange and perfect union he had with Ursula was dependent on the shared generosity that let each other be what they needed to be and be the best they could be. So paired with the remembering is the celebration, since that, after all, is the occasion tonight. But I first want to take a few moments to continue with remembering. My mother, Xiaowei Zhuang, who is 92, worked all her life as a biochemist. She was born in Shanghai and studied chemistry at Aurora College, a women's school run by the French Jesuits. I guess that's how she got the sort of nickname Yvonne. Anyway, she recently told me of having a story about having bed bugs infest the woven bamboo mats that covered the rope beds that she and her family slept on. She watched her mother pour boiling water on the mats, but nothing worked. So one night, she waited until the other students left the lab and proceeded to formulate DDT crystals, which she brought home to dilute and put into a spray gun and thus kill the bed bugs. So she was an imaginative and rather unruly problem solver. When she immigrated to America, she was a working mother and a woman scientist in the 1950s, when such a combination was very rare. I know she suffered through the issues which are still present for women working today, but she said what helped her get through was the generous mentorship of her bosses, who encouraged her and supported her and who were men. So I celebrate her because she is my role model. So what do I do? I'm not a scientist, I'm an architect. The work that I do with my husband, Todd Williams, and with our studio based here in New York, is primarily nonprofits and institutions. Simply put, we want to make buildings that contribute in some way to making the world a better place. So we've designed a public skating rink in Prospect Park, and it becomes free water play in the summer. We've created a new home for a legendary art collection called the Barnes Foundation in Philadelphia that makes this collection, which is an incredible collection of Impressionist art, accessible to a greater and more diverse population. And we've designed a home in Hong Kong for the Asia Society, an organization started by John D. Rockefeller to promote understanding between Asia and America. And finally, we are in the midst of designing the Obama Presidential Center in Chicago, which, unlike the traditional presidential libraries, will be devoted to the education of young people locally, nationally, and globally in civic engagement so that they can work effectively to make their worlds, our world, better. So tonight we're gathered here together to celebrate scientist Xiaowei Zhuang. After graduating from the University of Science and Technology in China, she went to gain degrees at UC Berkeley and Stanford, winning numerous distinguished awards for her research before joining the Harvard faculty. In trying to gain an inkling of understanding of what she is doing, I watched a lecture that you did on YouTube. And it's one she gave at Stanford, where she spoke about her research using the acronym STORM, or Stochastic, Optical, Reconstruction, Microscopy. I can't even begin to understand the depth of her science, but I could see immediately that she is a teacher who talks about her work with enthusiasm and humor and generosity. As the lecture progressed, we entered smaller and smaller worlds within worlds, and I suddenly had a sense of the beautiful and unending mystery of scientific inquiry. That was for me a tiny epiphany, so thank you for this gift, Xiaowei. While our work focuses on buildings, physical objects that are much larger than I am, Xiaowei's work is with particles that are much, much smaller. But strangely enough, I realized that we share the desire to make the invisible visible, to understand phenomena in 3 dimensions. Her work for the non-scientist, as you saw, has an exquisite visual beauty, which I think is not a byproduct, but is intrinsic to her research and understanding of the universe. So tonight, we celebrate Xiaowei, and we celebrate the thread as it continues, from Pearl to Paul to Xiaowei. The cloth that is made from that thread is warm and spreads wide. It nurtures and honors, and it is truly beautiful to behold.
Thank you, Billie, for those beautiful comments. I would now like to introduce Xiaowei Zhuang, our honoree this evening. Xiaowei is the David B. Arnold Professor of Science at Harvard University. She holds faculty positions in the departments of chemistry and chemical biology, and also the Department of Physics. She's an investigator of the Howard Hughes Medical Institute and director of the Center for Advanced Imaging at Harvard. Xiaowei was born in Hefei, China, born to parents who were both university professors. She grew up with their PhD students frequently in the home, and their excited discussions with her parents convinced her that being a professor must be a fun thing to do. She decided that she would become a professor. In middle school, she received formal instruction about gravity and other forces. She enjoyed using simple ideas like Newton's laws of motion to solve problems, in part because she didn't have to memorize anything. She could just figure them out from first principles. Her career goal then switched from becoming a professor to becoming a physics professor. In 1991, Xiaowei earned her undergraduate degree of physics at the University of Science and Technology of China, and then made a big jump, coming to the United States for graduate school at the University of California, Berkeley. There she did her PhD in the field of optical spectroscopy with the renowned physicist Yuanran Shen. She then, for postdoctoral fellowship, moved to the laboratory of another renowned physicist, Stephen Chu, at Stanford for postdoctoral fellowship. He encouraged her to use physics to develop new tools that would illuminate new areas of biology. With Chu, she began working on a newly conceived area of biology, single-molecule biophysics. Rather than trying to understand biology from studying the average behavior of thousands of molecules, these investigators zeroed in on examining the behavior of single molecules in real time, an idea that was over the top for its ambition 20 years ago. With impressive successes in this area, in 2001, she joined the faculty at Harvard and decided to look at biological molecules in their native habitat, inside the cell. This goal presented new technical challenges as the cell is crowded. She could no longer spread out the molecules one by one while examining them. Xiaowei soon realized that a significant hurdle impeded progress. The best microscope lens can focus light to illuminate a spot that's about 200 nanometers in diameter. Similarly, a fluorescently tagged molecule emits light and forms an image that's about 200 nanometers wide. But because biological molecules are about 10 to 100 times smaller than that, when they're close together, the signals of two different molecules can't be distinguished from one another. So to overcome this so-called diffraction limit, she conceived a new strategy. Rather than allowing all of the molecules to shine at the same time, she turned different subsets of them on and off, using dyes her lab discovered that could be activated and deactivated at will. This method, so-called stochastic optical reconstruction microscopy, STORM, allowed her to pinpoint each molecule's precise position, overcoming the so-called diffraction limit. Eventually, she sharpened the resolution to the molecular scale. She extended STORM into 3 dimensions and used it to discover previously unknown structures in axons, and we had an exciting conversation about this before tonight's talk about work that has extended on from that. Axons are the long fibers that conduct messages in the nervous system. The framework she unveiled serves as a platform for many molecules that work together to promote the signaling that fosters neuron growth and communication. STORM was a career-defining innovation, but this was not the end of her innovative ideas. For example, she knew that it would be useful to measure not only the quantities of messenger RNAs, but also to map them spatially within cells. Available methods could gather both types of information for only a few messenger RNAs at a time. Accordingly, she devised a technique called MERFISH, multiplex error-robust fluorescence in situ hybridization, to assess the activity and location of these gene transcripts at the whole genome level within intact cells. She then applied this technology to analyze nerve activity in the brain region that controls numerous essential functions, but in which roles of individual neurons were largely unknown. She continues to identify technical barriers and then just break right through them. Xiaowei is a member of the National Academy of Sciences, the American Academy of Arts and Sciences, and a foreign member of the Chinese Academy of Sciences and the European Molecular Biology Organization. This is how you know that everybody wants her and admires her work. She's also a member of the American Philosophical Society. She has received many distinguished honors in addition to the Meister Greengard Prize. In 2003, she was named a MacArthur Fellow. In 2019, she received an award for Scientific Discovery from the National Academy of Sciences. In 2018, she received the Heineken Prize for Biochemistry and Biophysics from the Royal Netherlands Academy of Arts and Sciences, and she won the 2018 Breakthrough Prize in Life Sciences. And now would you please join me in welcoming a remarkable scientist, Xiaowei Zhuang, to the stage. Billie, please come up as well. So you stand here. Billie, come over here. Congratulations. So please hold your applause. It's now my pleasure to read excerpts from the citation that is being presented this evening. Xiaowei Zhuang, the Rockefeller University is proud to honor you as the recipient of the 2019 Meister Greengard Prize. You're being honored for developing transformative tools in super-resolution microscopy, a technology that has illuminated the detailed inner workings of cells and has made deep impacts in diverse areas of basic and medical biology. With your new imaging technique, you broke through the diffraction limit, dramatically increasing the resolution of subcellular microscopy and allowing a completely new way of looking inside cells. You also devised a new method for monitoring the presence and location of thousands of messenger RNAs inside single human cells, which provides insight into gene expression patterns. By applying these inventions and others, you have unveiled unanticipated aspects of cell and tissue function. You have set the highest standard for the community of scientists by demonstrating how boldness, creative thinking, and determination can fuel discoveries with far-reaching impact. Today, in presenting you with the Meister Greengard Prize, we recognize and thank you for identifying and solving monumental technical problems and for applying your innovations to elucidate fundamental life processes that hold great promise for improving human health.
So it is my honor this evening to present to Dr. Xiaowei Zhuang the Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. On behalf of the Distinguished Selection Committee of the Meister Greengard Prize and all of us here today, we congratulate you on this award.
Wow. I now have to take a deep breath. It's really I'm so touched by this special and memorable event. And I'm truly deeply honored to receive the Meister Greengard Prize. So I want to take this opportunity to say a few thanks. And first, I would like to thank Paul Greengard and Ursula van Rijdingswaard for setting up this truly special prize to honor outstanding women scientists. It's an incredibly inspiring vision that Paul and Ursula had. So actually, ever since I received the call from Rick in March, I have had looked forward to this event. And I had looked forward to share our latest, scientific discoveries and discuss science with Paul. And I knew I would have learned so much from him, given that he is such a giant and expert in, neurobiology and our interest also in neurobiology. So this is also a pretty particularly sad moment for me, not able to discuss science and celebrate the prize with Paul anymore. So, Paul's science has inspired us all, and his passing is truly a great loss to the scientific community, so we will all miss him. I'm also deeply grateful to the prize committee members for choosing me as this year's awardee. And I looked up, as probably other awardees would have looked up the previous laureates. It's just an amazing list of extraordinarily accomplished scientists. And I feel incredibly honored that the committee choose to include me in this group. And as we all know, that there are just so many more great women scientists that then could be included in this list. So I also feel particularly lucky. Thank you. And I also want to take this opportunity to thank my mentors, in particular my PhD advisor, Ron Shen, and my postdoctoral advisor, Steve Chu, and both of whom Rick mentioned. And Ron, he taught me how to think independently and critically, how to question conventional wisdom and arrive at my own conclusions. And Steve, he encouraged me to follow my own passion, to inspire me to form a exciting vision. And I still remember when I just joined his lab as a postdoctoral fellow, I just said, Steve, I want to work on something new. By new, I meant neither you nor I have worked on. And he said, great, I got interested in biology. Why don't you look into biology? Even though I was a pure physics major and I pretty much know nothing about biology. So it's really amazing that Steve, he doesn't think science should be bounded by disciplines. And he believes that everything is possible if we put our hearts and minds to it. So these great scientists, their styles have influenced me tremendously in my own approach to science and in how I interact with my own graduate students and postdocs. And I do have to say that working with these talented and dedicated young students and postdocs over the past 18 years has just been the most rewarding experience that I've had. And I'm glad that one of them is here joining us today as a representative, and that's Shixing Liu. And he's a faculty member here at Rockefeller. And it is because of Shixing and other students and postdocs like Shixing, I could stand here to receive this prize. So I feel that this is a celebration for their accomplishments as well. And I would also like to extend my thanks to my wonderful collaborators. Several of them are right here at Rockefeller. Titia DeLong, my dear friend, Rod McKinnon, and Mark Tessier-Levine, who used to be here at Rockefeller and now at Stanford, and many other collaborators in other institutions. As we all know, science today has becoming increasingly more collaborative and multidisciplinary. So I feel really lucky to have the opportunity to collaborate with these great scientists, which made the journey to discovery just that much more fun. So importantly, I shouldn't forget that. I would also like to thank my family and my loving husband, daughter, and parents. They've been so supportive. They've given me so much joy in life, and they've accompanied me through life's ups and downs. And importantly, they all indulged me, particularly indulged my strange hour working habits and so on. And my daughter, particularly for indulging me with all the travelings that I do. And then she just, when I say, Iris, I'm going out of town. Okay, I knew that. So she's lovely. And finally, last but not least, I would like to thank my friends and colleagues here at Rockefeller University for putting together such an amazing event. And thank you, Rick, and thank you, Billie, for your wonderful, beautiful presentations and generous presentations. And as some of you know, Rockefeller is my favorite place. And every visit is like a real treat to me to learn about the latest scientific discoveries from the scientists that I admire. And today, you make me feel really special. Thank you.
So we'll now have a little time for questions and answers. I'll start, and if there's time toward the end, perhaps we'll take questions from the audience. So first of all, thank you for your beautiful remarks and congratulations again. Perhaps I'll start by just asking what you were thinking about as you, as a trained physicist, started looking at biology. How foreign did biology look to you, and how did you find your way to problems that you thought were solvable?
Yeah, so it is very foreign to me. So I used to say this, and nobody believed me. When I started as a postdoc in Steve's lab, I didn't know the difference between RNA and DNA. So that is really remarkable. You know, I did not take a course in a biology course in college. You know, as a pure physics major, they didn't think it was necessary for me to take biology course, and my biology stopped at high school. And high school back then in China doesn't get to molecular level. Of course, I know DNA is a genetic material and RNA is a messenger. What I meant is I don't know the difference, the structural difference between them. So that really made biology incredibly foreign to me. But in the meantime, I felt like I also didn't know that it's a difficult subject, and there's so many challenges there. So when I was told, why don't we look into biology by Steve, I said, oh yeah, why not? Sounds good. So it also made the transition sort of easy because I was ignorant. Ignorance is a bless sometimes, right? So yeah, so that's how I got into biology and how foreign it was.
So you're not the first physicist to come into biology. About 80 years ago, Max Delbruck was a German physicist who, in fact, on a grant from the Rockefeller Foundation, came to Caltech to start working on what became the revolution in molecular biology. And it's said that he was prompted to do that on advice from Niels Bohr, and, which raises the question, there's a nice parallel between Niels Bohr, a great chemist and physicist, telling him to go study biology, and Stephen Chu giving you the same advice some 80 years later. At that time, people were coming in from physics looking to find new principles of physics in biology and life forces. But you took a rather different approach, thinking about how to overcome barriers. So this gets to my question. I would love for you to describe what interests you in science now, asking this audience not to hear Xiaowei talk a little bit about science is like inviting Pavarotti and asking him to come play tennis for us. So I would like to hear your thoughts about perhaps how you hit upon the idea of studying the diffraction limit and solving it, and what excites you in science now.
Yeah, so actually you said quite a lot of that in your speech. When I was a postdoc, I did all in vitro studies, meaning study molecules in solution and so on. And then when I started my own lab at Harvard, I thought, these molecules, they don't live in solution. They actually live inside our cells. So I want to see how they behave in living cells in their native environment. And there, pretty soon I encountered the diffraction limit. To give you an idea of what it is because if you have a molecule that emits light, and because of diffraction, the image has a finite size of 200 nanometer. Now, 200 nanometer, how small that is, so that's about 1/100th or 1,000th of a hair width. You think that's small. But the molecules in our cells are even smaller, 1 or 2 orders of magnitude smaller. And they form these structures that if you have the diffraction limit, these structures are just blurry. Using an example, when I started at Harvard, we started to look at how viruses invade cells. Now, viruses, they're made of a lot of different amazing molecules. But if you look under the optical microscope, it's just a blob. And how they invade cells, they often take the cellular machinery to do that, such as clathrin-coated pits. That's also made a lot of interesting molecules. And then how do they interact and invaginate and then take the virus into the cell? But if you look at clathrin-coated pits under the optical microscope, it's still just a blob. So, it made these amazing molecular machineries just all into unresolvable structures, which is very frustrating. So because of that frustration, I just have the desire to overcome that. And then having some physics knowledge is helpful because I know what's the basis, what's the problem there. What's the key problem is because each molecule emit and give an image that's 200 nanometer in size. When they're close, they overlap. The overlap is the key. So in order to solve the problem, you have to solve the overlapping problem. And then everybody needs a little bit of luck. We got lucky because we accidentally, serendipitously discovered photoswitchable dyes. And then we realized there lies a solution, because I could then use these photoswitchable dyes and activate only a subset so that their images no longer overlap. And then I can pinpoint their center position. And then we can iterate this process, determine all the molecules' positions with nanometer positions and the precisions, and then I stack them together, then we get a super resolution image. So
Easy.
I mean, it's true that it might sound easy, but it did take us quite a while to actually put it in practice and to make it work. We had to overcome a lot of, some of the barriers, not even what you imagine, because when we had that, it was not the method itself is difficult. It was like, well, now you have this better resolution. How do you prove to people that your better result structure is real? So we have to engineer structures with known shape and so on. That took us even longer than actually putting together the STORM microscope together. But it's really wonderful now that, I mean, also I do want to say that STORM is not the only super resolution imaging methods. There are other super resolution imaging methods too. And together, it's actually a very big field and a lot of activities of both biologists and physicists. And together, because of these efforts, we can see structures inside the cell with just much better clarity. And there's an explosion of new knowledge in cell biology because of this. So it's really thrilling to see that.
So, the mark of somebody who is a technology developer is not that you developed a cool technology, but that everybody recognizes how useful it is and immediately adopts it. And the development of super resolution microscopy is one of those developments that everybody, including here at Rockefeller, we have multiple super resolution microscopes. And Allison North here runs a facility that specializes in training and maintaining these instruments. So doing that once is remarkable. Doing that a second time in a rather different arena is even more remarkable. You want to say a few words about
the
what
MERFISH?
MERFISH does?
Yeah, so MERFISH came out a little differently. It was not much because of a frustration. It was, we were talking about, Rick was asking me, oh, what's new these days? Do you have a brand new method and so on? Well, sadly not yet. But when I thought of MERFISH, it was because at the time, the STORM has becoming pretty mature, and we're applying it to make biological discoveries. And then I felt this itch of having not just incrementally improving some methods, but have an entirely new method that can break new ground. And it's also at a time that you hear a lot about the next-gen sequencing, the single-cell genomics. People can, look at things at the genome scale with high throughput. You get lots and lots of information. And then I thought, wow, wouldn't it be wonderful if I could do that by imaging? And of course, the first thought is a bit naive because it was just because I love imaging. As you can probably see from the video, I really like this direct visualization approach. But then think a little bit more about it. It makes sense by doing it by imaging because to skip you a little, one particular problem. Our body is made of many, many different kinds of cells. And these cells are different in their behavior and function. Not because they have different genome, it's by and large the same. It's because they have different gene expression profiles. And then if you could actually look at all the genes, their expression profile at once, then you can identify these different types of cells. But if you do that by imaging, you don't have to take the cell out of its native environment or the native habitat, and then you could actually get the spatial organization. You could totally understand why the spatial organization is important, because if you mix all the different types of cells together randomly, just imagine what we would look like, a blob of no feature, right? So apparently the spatial organization is key to the function of how we are living beings and so on. So if I could do that by imaging, identify the cell types, see their organization, see how they interact. That would just tell us a lot about biology, fundamental biology, and diseases. So then I thought, well, it is great to dream about doing it by imaging. And how is, of course, the question, because we're talking about imaging thousands of different genes. And I've been working in imaging for a long time. I know that if I want to image the 2 different kinds of genes, I do 2-color imaging. If I do 3 different kinds of genes, I do 3-color imaging. If I do thousands of different genes, I don't have 1,000 distinguishable colors. And I also know that if you do one at a time and then stack it, my students would hate me. So I'm trying to think of a way that is efficient to do that. And then it hit me. It is the combinatorial approach. So basically, you image a group of genes, different group at each time, an overlapping different group. This way, I can actually image the number of genes that I can distinguish grows exponentially. What's the number of rounds that I image? This doesn't last long enough for me to explain the details. I'll be happy to do that if afterwards there's time, but just trust me that then what means this exponential thing means you can actually image, say, thousands of genes with just, say, 20 rounds of imaging. That seemed to me manageable. So I just told my group this idea. Everybody loved it. And soon we found that it needs an additional element, and that is if you do this in our technical way of saying it's 20 rounds of imaging, 20 bit of imaging, every bit has a little bit of error, but they add up. And then we actually then thought, oh, we can use codes that can detect and barcodes that can detect and correct error to solve that problem. So that's why we call this multiplexed arrow busfish. And nowadays we can use that to image really thousands of genes, their expression profile in individual cells, in the native tissue environment, to identify new cell types, to map out their spatial organization, their function. Basically, you can imagine what you get is a detailed spatial, molecular, and functional atlas of cells in your body. And of course, it helps understanding fundamental biology, but we also envision that it could potentially help disease diagnosis and precision medicine one day. So
terrific, thank you. So, so as a woman in science and a woman particularly starting in physics, fields that are not known for being the easiest fields for women to move into, I'm wondering if there are young women scientists in the audience. I'd be interested in your advice to them.
Yeah, so my advice is, just do what you love to do and believe that if you try hard, you will succeed. And there is fundamentally no reason to believe. We know that men and women are different. They're different in, it's stupid not to admit they're different. But there's no reason to believe that the difference would make one gender better at science than the other. So we really can all do extremely well if we try hard. And maybe then just when you're doing science, just don't think that you're a woman or man. We're all just scientists. And we do it because we love science, and we work hard because we want to make discoveries that help the society and so on. So put your hearts to it, and you will succeed.
Thank you. We've got a few minutes for questions from the audience. We have roving microphones, so if you have a question, raise your hand and a microphone will come your way. So a question here.
Thank you so much, and I have to say I'm incredibly impressed by the way your research is really technical, but the way you explain it, like I could understand it if I knew absolutely nothing about it. So I was wondering, how did you learn to communicate science so well, and how do you think we can teach and encourage better science communication?
Yeah, so how did I learn? I'm not sure how I know how did I learn. I just have a desire to make things clear to people. And then I learn also from people who give me feedback. You know, for example, I actually don't know whether I used this word, you know, when it's MERFISH. Did I say that the word genome scale? And when I was talking to the person who's writing up the work about writing up about our work, it's Evelyn Strauss, and then she told me that genome is not necessarily a word that everybody knows, so you have to explain that clearly. So then I think today I just explained thousands of genes. Now maybe gene is also a word that is too technical, then I don't know what to do. But I just like to make things clear to people. You know, I think it's just a desire. Actually, there is an interesting anecdote. I was giving a talk at Stanford after I got a job offer at Harvard. It was not a job talk. And then I gave a talk, and then a fellow postdoc, at that time I was a postdoc, a fellow postdoc came to me to say, Xiaowei, your talk is so clear. It perhaps is not good for you to give such a clear talk because people think what you do is trivial. He said, it's good to make it a bit mysterious. And I just, it didn't resonate with me. I mean, I like people to know that, I didn't have the chance to explain to my grandmother what I do, but I do explain to my mother what I do. So, you know, being able to explain and have people to share your excitement, joy, I think it comes from an inner desire. So I didn't particularly practice that, but I do practice my students for that. So I ask them to give practice talks and pointing out where it's unclear and how to make it clear. So, and throughout the, years in their lab experience, they get better. So it is a skill that can be acquired.
Other questions?
Hello. Well, first off, congratulations. It was really amazing to hear about your work. My name is Yvonne Chow. I represent a dance non-governmental organization called H+, the Hip Hop Dance Conservatory. And you're probably wondering why I'm here. Our tagline is guided by science, powered by soul. So we consider ourselves kind of like the nerds of dance. So this was just really impressive. I wanted to know, because I know that you have students that you work with and things like that. What do you see as like one of the common challenges that young scientists or just scientists in general face, and that you think can be addressed, in sort of like an internal way? Like, are there things that you see that come up over and over again in the science community that you think could be different?
That's a tough question, actually, because, I'm sure that there is challenges when it comes to communicating science to people who are non-scientific, to make them realize what we do are important, to make, people trust this long view that the society should support, long-term science work, even though it doesn't necessarily pay off immediately and so on. And, sometimes we all have to sort of being willing to be lonely and then do things, and that takes a long time to see its impact. But I say it's tough is because we don't really experience that kind of challenge every day, because we are nerds, but we talk with nerds. We communicate with people in our community. Everybody, are scientists, and we appreciate each other. So we make each other feel good. So was that? I don't know whether I'm answering the question well. It's a bit of an unexpected question, but to me, I think science is a lot of fun. It's true that nothing always works. Everything is, what I meant is not nothing always works. What I meant is things do not always work. You will experience challenges and so on. But, every once in a while, we do have great success, and that keeps us going. And also the fact that we love to understand, then even failure doesn't really make us feel miserable because we still understand from failures and so on. So all these things keep us going. But sometimes it's frustrating if you talk to people who are a little bit more remote and not in the scientific community, and then they look for more immediate, things. They ask, what do you cure cancer tomorrow? Things like that. And I'm a bit of exaggerating, but sometimes you do feel that that kind of sentiment exists outside the scientific community. But if we all take that kind of a short view, then a lot of great things that today did help you so much would not have happened. These days people take certain things for granted. I give one example, MRI imaging, right? And it's one of the most powerful diagnostic tools. But then it came from this physics thing called nuclear magnetic resonance. Well, then when the person discovered nuclear magnetic resonance, he would have no way in imagining in the future it has such a strong impact on our human health and society benefit and so on. So it is that kind of a thing that we do that someday, maybe 20 years from now, maybe 50 years from now, maybe 80 years from now, will help the human society. And the people who are, the general society people and the politicians and so on, they need to understand a bit of this kind of a long-term investment and our curiosity-driven research. Not all of them will eventually be impactful, but even just a 1% or 0.1% to have the kind of an impact as MRI, it's all worthwhile. So.
I think that's a good note to end on. Thank you again, and congratulations, Xiaowei. It was really wonderful. Thank you so much for your terrific comments. That was just great.